Going to get into our webinar. So this year, through the Hawaii Invasive Species Awareness Month, this is an annual event dedicated to educating and mobilizing the residents of Hawaii to protect our islands from the threats posed by invasive species. This year's theme explores what's at risk, because we often find ourselves immersed in learning about the next invasive species threat. But this year, we wanted to shift our focus onto the beloved native species, their ecosystem functions, and the cultural practices being threatened. So awareness and compassion is critical for invasive species work, because without those things, it's impossible to get people to care, to observe, and to make change. So this morning, we will get to hear from two leading insect researchers in Hawaii who will take us on an exploration into the hidden lifestyles of Hawaiian insects. From bone collecting caterpillars to camouflaging spiders, we'll have everything in this little webinar. So I have an opportunity for discussion at the end of our presentations. So if you can please remember to send over any questions you may have through either the Q&A feature, which you can find on the bottom black bar, or through our chat feature, whichever is easier for you. All right. So I think with that, we'll get started with Dr. Camille Doorenweerd. You can start putting your slides on when you're ready, and I will give a little introduction. Okay, so Dr. Camille Doorenweerd is a research faculty at the University of Hawaii at Manoa, where his main research focus is on the systematics of invasive species with economic relevance, like fruit flies of the tribe Dasini, that threaten agriculture both in Hawaii and the mainland United States. As one of a handful of professional entomologists on in the Hawaiian Islands and trained on taxonomy, he is also closely involved in various conservation efforts, ranging from ecosystem restoration to getting baseline biodiversity estimates for land management purposes. Dr. Doorenweerd has a broad interest in evolution and in particular to speciation, which in Hawaii is inevitably intertwined with conservation research. Dr. Doorenweerd is a curator of the University of Hawaii Insect Museum, as well as the treasurer of the Hawaiian Entomological Society. So mahalo nui for being here, and we're super excited to hear your talk today. Thank you.
All right, Darcy, thank you for that introduction. So yeah, my talk for today I've titled Fantastic Hawaiian Insects and Where to Find Them, and I hope to take you on a little journey into the wondrous world of Hawaiian insects. Let's start by putting things a little bit in perspective with some biodiversity numbers. Let's talk specifically about the endemic species in Hawaii. So those are species that are only in Hawaii. They evolved here and they're not found anywhere else. So these are really our most treasured species, if you will. And that's also what we stand to lose if we don't take care of them. So you've probably heard, most people have heard of the endemic birds. So this is not all the birds, this is just the endemic species of birds. There's 44 species of endemic birds remaining, which is an amazing diversification, especially of the honey creepers that most people know about. But much more diverse than that is already the snails. There's about 767 species described right now. They're still discovering new species. So this is an immensely diverse group of organisms on the island. But even more diverse than the snails is the plants. There's about 1,260 endemic species known right now, and they're still discovering new species there too. So between these three groups, you've already got over 2,000 endemic species. So that's just a beautiful biodiversity. But as you may have guessed from the amount of white space that's still left on this slide, the insects are even way more diverse than all those, the birds, the snails, and the plants together. So by our best estimates, there's about 10,000 endemic insect species on the Hawaiian Islands. So that's insects and spiders, I should say. And of those, only about 5,000, a little over 5,000 have been described. So about 50% of the diversity here hasn't even been discovered and named yet. So there's still a lot of work to do. And what's interesting to note here is that when you're talking about conservation funding, it's kind of the inverse of the diversity that we have. So most of the money, millions every year, goes to the conservation of birds, and much less than that goes to snails and plants. But at least there is a snail extinction prevention program, and there is a plant extinction prevention program. There's no official insect extinction prevention program. So really, even though they're the most diverse group, they're getting the smallest cut of the conservation funding. And I think in part that is largely because the same reason that made them so evolutionarily successful, and that is that they're really good at hiding. And a lot of them are really small. So people don't really see them and they're not as familiar with them. So I'm really glad I get to talk here today and hopefully spread some awareness about how cool they are. So I can't talk about all 10,000 of them. That's too much for 20 minutes. My main passion is about the moths. So that's about 2,000 species. That's still too much for this talk, so I will narrow down more. And here's one of my favorites. So I have many favorites. Actually, it's really hard to pick my favorites, but this is one of my favorites. It's Plutella noholio. It's only found on Hawaii Island. So let's start with where can you actually find native insects before we move to the how. So where can you find them? You can find them on any island from sea level and all the way up to the top of Mauna Kea. There's the vacuum bug up there. But you'll find the highest diversity of insects in areas where native plants are abundant. So this is a little video. The video work is not great because that's me, but you see our the PI of our lab, Professor Dan Rubinoff, with his son walking in the boardwalk in Kokee on Kauai. And you can see that this is just beautiful, varied habitat. There's lots of native plants. There's lots of structure, like there's canopy, there's open spots, and that's really this mesic forest, so this wet forest, that's really where you can find the highest diversity of insects right now and also the highest abundances. But if you look hard enough, you can find them everywhere. So that's where you can find them. So the other tricky part is how you can find them. And today I'll be talking about two groups, two of my favorite groups among the many favorite groups. One is in the genus Philodoria, and the other group is the genus Hyposmocoma. So these are tiny moths, right? So the scale bar is down here, but they're also beautiful. Philodoria, there's over 50 species described. There's more undescribed species, and this whole, what we call is a radiation, is completely endemic to the Hawaiian Islands. So that means that at some point, millions of years ago, long before Kauai even existed, an ancestral species arrived here, and from there, over the course of millions of years of evolution, it specialized on different resources and speciated to become all these different species. So Philodoria has over 50 species. And then this other group I'll be talking about is Hyposmocoma. That group is just completely crazy how diverse it is. Right now, there's over 400 species known, and that number may actually double because they're so diverse. But let's start with Philodoria. So the first step into finding Philodoria is to know a little bit about them. And one of the things about them is that they're very host specific. So you need to know a little bit about the Hawaiian plants to know where to find these moths, because the moths specialize on certain plants. So the names I've put here is the names of the plants that the moths can be found on. So there's a species or several species that can be found on ohia. There's several species on Kolikole lehua, mamaki, kolea, and kokio. It's just some of the examples. There's other plants too, of course. But that's kind of the first step to finding them, is knowing a little bit about the plants that these insects are associated with. But you may notice all these scale bars in these photos here. These are really tiny moths. So 5 millimeters, that's roughly a quarter of an inch, right? So this is very small. So you may ask, like, okay, this is great, I can find these plants, but these moths are just gonna look like mosquitoes to me. So how do I find them? Well, there's a trick for that too. So this is a video. I'm only going to show a short fragment of it, but it was made by Chris Johns, who did his PhD on Philodoria between like 2015 and 2020. And he was really good at finding them in the field. And he was also really into macro photography and videography. And he actually got a grant from National Geographic to make this video. And this is Chris, and here you can see quite how small these moths are. But the main reason I wanted to show this little clip is going to come up right about here. This is Chris telling about his work. I really recommend you, the video is for free on YouTube. I recommend you watch it. But this is what I want to show you. So this is the caterpillars eating. So this is a time lapse over the course of several days. So in reality, it goes much slower. But these caterpillars are called leaf miners. So they are so small that they fit between the upper and the lower epidermis of a leaf. And as they feed, they create this little corridor, and that's called a leaf mine. And so they eat away through the leaf until they've eaten enough and they're big enough. Then they exit the leaf mine and they go into the soil to pupate. And then out comes this beautiful little moth. So here's some more pictures of what those leaf mines look like. I think this is all Mersini. The arrows here, they indicate where the caterpillar is at. So slowly as the caterpillar feeds through the leaf, it grows and the leaf mine gets bigger. And that's actually not that hard to find when you know what you're looking for. You just have to take the time. So of course, when you're hiking at 10 miles an hour and you're just racing through the forest, yeah, you're gonna miss them. But if you take some time and just, just be in a place and just look around and just stare at a bush for a while, that's basically what we do. And you'll start seeing them. And as your brain gets trained into these search patterns, you'll actually start seeing them a lot. So it's kind of a blessing and a curse, but it's really cool. And always when I tell people this, there's some people in the audience that are like, yeah, I have these too in my backyard, and that's totally possible, especially if you live in Volcano or something. But there's also other groups of insects that make leaf mines. It's not just Philodoria, including introduced leaf mining flies, which are probably more common around your house, especially in urban areas. So one way to tell the flies from the moths is that if you look at the leaf mine with the moths, there's all these little pellets in here. So that's the poop. We call it frass, is the technical term. And it's little pellets, right? So if you look at the fly leaf mine, doesn't have the pellets. It's just kind of like diarrhea in there. So you can see it especially well if you hold a leaf mine up against the light. So if it has little pellets in there, it's probably from a moth, and otherwise it's probably a fly. So that's Philodoria. Moving on to Hyposmocoma. So Hyposmocoma, like I mentioned earlier, is just amazingly diverse. Like I've studied moths all over the world, and I haven't seen a genus anywhere that is as diverse as Hyposmocoma. They're diverse in size. Like the smallest ones here, you can see this is a 1/4-inch wingspan, and then there's others that have a wingspan of about an inch. The wing patterns are highly diverse. Their ecologies are highly diverse. It's just absolutely amazing what happened here. But again, these are tiny little moths, right? So how are you going to find them? And also, again, the answer is that you actually look for the caterpillars, although in a different way from Philodoria. So this is a little video I took while I was staring at a tree trunk, because that is what you do when you're looking for Philodoria. And basically, this tree trunk had all bits of lichen on it, and you're looking for bits of lichen that seem out of place. Or in this case, this little bit of lichen started moving around, and lichen does not move around. So what we're actually looking at here is the larval case of a Hyposmocoma. So here you see another species where it got flipped upside down, but that's basically the same thing. So the caterpillars are tiny, but they built these little houses from silk that they carry around, like a snail carries its little snail house around. And it's really cozy, especially when it's all decorated with lichen like this. And then the little opening it has there, it can actually close that up too. So then it's just got this amazing little shelter that's built for itself. And all the Hyposmocoma do that. And they make different case types. So the cases can look different, and it helps to identify the different species groups already in the field. So we made this Hyposmocoma case type field guide. If you want a copy of that, it's available on our University of Hawaii Insect Museum website. You can have it on your phone when you go out in the field. You can see what type of cases you might be able to find. We gave them all these funny food-related names. That was mostly our PI, Dan Rubinoff. I have to credit for that. But it is very helpful to get you as a first step towards identifying what species you're looking at. And then I have a little video here. Yeah. So this was a rock that was, it's got several cases on it. So at the bottom here, there's a candy wrapper. My camera work is not great, but there's a candy wrapper. This is a bone collector right here. This is a flat purse. And then on this side, there was actually a purse as well. So we had 4 different case types just on this single rock. So a lot of these are actually not that rare once you get into decent forest with some native plants. You can find a lot of these. So I do have to mention too, like if you do find them, don't take them. Just take photos or enjoy them where they are, because all native insects require permits for collecting. So just take photos and enjoy them as they are. But do try to find them and take those photos, because there's only so many entomologists and we cannot find everything. So all the information we can get, you know, posted on our naturalist, that's all helpful for our research. And again, I have to mention here too, like there's always those people who say like, oh, I have these all around my house. And it's possible, but there's also some non-native species of which the caterpillars make cases. So there's the bagworms, for example, especially if you don't cut your grass very often, you'll have a lot of these. And then there's the clothes moths relatives. It's kind of similar to the flat purse, but you can see the flat purse has this curved edge on one side and then a squiggly or wavy side, whereas the non-native species has a wavy side on both sides, so it's more symmetrical. But yeah, we've got all these different case types, so flat purses, crabs, and it tells you a little bit about their ecology too. So crabs are mostly feeding on fungus, cigars are carnivores, and then you may have heard of the bone collector, which is also a carnivore. So the bone collector, we published about that a couple weeks ago in Science, and it got a lot of media attention, which is great because that means more attention for Hawaiian insects. But it was actually, we found the earliest ones in 2008, but it took us some time to figure out what was going on, because at first we thought, like, oh, it's just randomly got some insect bits on there. And then over time, as we were finding more of them, we realized that they always have spider parts on them. And then we were finding them more and more with spiders too. So we started to realize that this is not a coincidence. They're actually using those parts, like it's usually the molts from the spiders. So we're not thinking that they're killing spiders. They're not that ferocious, although they'll eat whatever they can. There's still going to be predators that are stronger than them. But they'll use the molts from the spiders to decorate their cases. And in that way, the spider won't recognize them as a threat while they're actually hanging out right by the spiders, which is probably a pretty safe place to be. This is a photo that we found, like this was a log. So they're usually in logs or under tree bark or under rocks. And this is a non-native spitting spider with its little keiki. And it seems completely oblivious that there's a caterpillar hanging around right there next to it. So that's pretty amazing about the bone collectors. But of course, I have to include this in this talk. I can't all be positive because we're talking about invasive species here. So we know very little about insects, right? As I started, only 50% is discovered, let alone that we know all of their ecologies. And we're slowly discovering more and more about how amazing they are. And you would really need to know all those details to firmly identify threats. There are some introductions like termites. We think termites have been really detrimental for especially a lot of Hyposmocoma because they traditionally would have been the decomposers. So there's direct evidence for competition at the very least. And then especially low elevations. In the early days of biocontrol introductions, they didn't do off-target testing. So a lot of the parasitoid wasps that were introduced for agriculture, they're also hitting the native insects. But if I have to talk about like really what's like a blanket big threat right now for native insects, it's still habitat loss. But it's what we've turned an invisible type of habitat loss. And it's not invisible at all if you know what you're looking at. But for many people before, like when they were filling up the wetlands and turning it into urban areas, when they were cutting the dry forest, turning it into agriculture, that's very visible, right? You move from a situation where you have a lot of green on one side and then a situation with concrete. Like you've clearly lost a lot of habitat. But the more invisible threat that we've moved into now is that green is replacing green. So to many people, this picture here might actually look like a pretty nice forest, like it's lush, it's green, but it's actually a forest that's on its deathbed. This is Upper Makawa on East Maui, and it's really sad to see this because what's happening here is the Himalayan ginger is completely taking over the understory. And you can still see the old growth, ohia and koa, but their seedlings just have no chance of getting through this. And they're probably still in the seed bank, so there is still time to save this, but time is running out, and this is happening really fast. So this ginger was not here 20 years ago. And sometimes we have discussions in our lab about what is the worst invasive, and of course we never agree on it, but the Himalayan ginger, especially for mesic forest, is always in our top 3. And then together with the pigs, of course, because they work in cahoots. So the pigs, they clear everything, and then the ginger just blankets over it, and then nothing can beat the ginger. And that's also a threat for all the islands, right? Because every island has its own specific threats too. But if we had to pick one thing that's really threatening major habitat loss or causing major habitat loss for the native insects right now, it's the Himalayan ginger. So that's definitely one that we would like to see addressed way more if possible. But there's a lot of initiatives happening and a lot of work is going in the right direction. So hopefully by raising more awareness and getting people more aware of that, this invisible habitat loss, I think we can get a lot done. This is just a little shot of our lab looking for Hyposmocoma and including the bone collector. So this is basically what it is. This is not even the best forest here. There's not a whole lot of native species, but it's still a really good spot turned out to be for Hyposmocoma. So just, yeah, my advice is take some time when you're hiking, just sit down or stare at a tree trunk, just stare at some moss on a rock, and maybe these beautiful moths will become apparent to you. All right, that's my presentation, and I'll gladly take any questions.
Wow, that was awesome. Like, wow, I learned so much, and it was so wonderful getting to see like the different videos of those little moths. Oh my gosh, they're so cute. I just had like one question before we and then I'll introduce our next speaker. But I was just wondering, like, so do the colors of the wings correspond to the plant that they are hosted on?
So that's one of the things we only know details of. So for the most part, we do think the colors are for camouflage. Especially the nocturnal ones. Most are nocturnal. So during the day, they're just trying to hide really and blend in with the environment. But then there's some that are really metallic, and they're probably day active actually, and they might try to be seen as part of a sexual selection. But that's all details where we know very little because we're still discovering species and we're very in the early phases of learning about their ecology.
Yeah, thank you so much. All right, so we're gonna have time for a Q&A after our next speaker, and then I will start getting us into that. So Natalie, if you wanna pull up your slides, and I will introduce you. So we have our next speaker is Dr. Natalie Graham. She is a molecular ecologist and assistant professor of biology at the University of Hawaii at Hilo, where she leads the ʻElala Biodiversity Lab. Her research focuses on Hawaiian arthropods and how human activities are reshaping ecosystems. By analyzing DNA from specimens and environmental samples, such as soil, water, and leaves, she uncovers patterns of biodiversity and species interactions. Dr. Graham's work is crucial for understanding how ecological communities evolve over time and how conservation efforts can protect Hawaii's fragile ecosystems. She has contributed to species identification research using molecular methods to help clarify the classification of organisms in Hawaiian biodiversity. With a deep respect for the Aina, she combines scientific discovery with applied solutions to safeguard native species from threats like habitat loss and invasive species. So with that, I will hand it over to you, Natalie.
Thanks. Hello, everyone. So we're going to talk today a little bit about the specialness of Hawaiian spiders. I really enjoyed listening to Camille's talk, and it was a great setup for thinking a little bit about biodiversity and insects and their kin. And so spiders are a group of arthropods with a lot of diversity in the Hawaiian archipelago. And part of the reason why there's so much diversity is because we're so isolated, and there's been a lot of time for evolution to act on the colonists that arrived millions of years ago. And this also has provided, because the islands are set out in this chronosequence, this kind of time machine to look backwards in time to understand what species arrived earliest and how they kind of hopscotched down the island chain. And so islands really provide this very simplified system for understanding colonization history, species interactions and how they're changing, the process of speciation, and of course the process of extinction. And so really what we're looking at is kind of this pairing interplay between ecological and evolutionary processes. And that's what a lot of my work is focused on, is kind of at the community level. But today I want to take you through a story of spider diversity. So this is actually just a snapshot of molecular phylogeny of spiders just at Kokei State Park. And again, just one sampling event at 6 sites in the park in the wet forest. And it's nice because it's representative of all of the different major radiations or large groups of spiders. So we have the Thomisids, which you think of as crab spiders, Philodromids, which people call running crab spiders. So you see the similar way that their legs are laid out, and they often move sideways. And then salticids, which this is your jumping spiders. There's a large number of these as well. The Hawaiian ones tend to be somewhat drab in color and not as pretty as some of the mainland ones. And then, of course, you're probably familiar with the happy face spider. Happy face spider are actually just one in a large diversity of theradiids. So there's Ariamnes spiders. This spider right here is Rugathoides, the triangle, very triangle shaped. And this is a new spider that we're just finding on Oahu and on Hawaii Island. And it has kind of an unusual body shape, and we're really not sure what the ecology is yet. Another group are these web builders that Araenidae, and then I'll be talking about them in talking about Cyclosa. This is a group that I worked on a lot. And then this is Orson Welles. So if you've ever seen large sheet web in the forest, these big flat webs, that's where this spider comes from. And it's actually a case of island gigantism. So on the mainland, they're really small. And then when they got to our archipelago, they got really big in body size. So their body itself is about the size of a quarter, and then their legs can be bigger than that. And what's interesting, actually, is that there's a whole bunch of undiscovered linyphiids. And so the reason why this picture right here kind of came out early is we thought that it was a theradiid, like the experts thought that it was a theradiid at first. And then we were able to use DNA to kind of place it within this radiation. And since then, we've gotten a grant to go back and kind of look for more of these, because it's a major undescribed radiation of spiders here. All of them are quite small. They live kind of underneath the bark of trees. And And so, yeah, just a lot of undescribed biodiversity in all of these groups. The big pink group of taxa at the bottom are Tetragnathidae. So Tetragnatha spiders are a huge, what's called adaptive radiation, where the ecologies have varied a lot as they have kind of moved down the island chain. I want to also point out that in this snapshot of spiders that we collected at Koke'i, there are some groups that are also entirely introduced families that don't belong here. And they're kind of whittled their way into our beautiful spider radiations. So for spider diversity, it really comes alive at night. So if you're saying, well, I've never seen half of those, that's probably because you were looking at the wrong time and not necessarily the wrong place. So just as the sun is setting and it's getting darker in the forest, you're likely to see one of these beauties just dangling down like little gems, like ornaments hanging off of the trees. And a lot of these spiders, that's when they go out to hunt as cursorial hunters. And so you'll really will just see them roaming around like little gems in the forest. The Tetragnatha spider are what's called an adaptive radiation, which means that there is some type of ecological diversification. That means that the way that they do their living varies quite a bit and is dependent on things like their diet or where they live in the substrate. And so part of this is that there's about 60 species, but there's actually more that aren't yet described as well. That have these wide variation in colors, shapes, sizes, behaviors, etc. And they're all found across the archipelago, and many of them are, localized just on a single island endemics. They have diversity in their webs, and Tetragnatha on the mainland are all web builders. You don't ever see a cursorial non-web building Tetragnatha. And actually, if you find Tetragnatha on the mainland, you will find them in a riparian zone. They build their webs over creeks and streams, and that's how they catch their prey. They build these somewhat flimsy webs and are kind of waiting for insects to emerge from the stream, and that's how they make their living. When they got to Hawaii, they actually moved away from the streams, and so there's web builders that are in any part of the forest. But there's also this other distinct clade, which is the so-named spiny leg clade. And these are the cursorial hunters. And there's some interesting things that's going on with cursorial clade. It's just so cool. So part of what happened is that each time that they hopscotched down to a new island, they generated the same set of ecomorphs. So if you got to a new island, you were likely to become this green ecomorph, or it's a different ecological type. You're also likely to become the maroon, which lives mostly on moss, small brown, which lives on trees, and large brown, which lives on tree bark. So these are the ecological affinities. It has to do with where the spider dwells. And every time that they colonized a new island, they evolved the same set of ecological affinities. So you might think that the green spiders are all the closest relatives to each other, or the maroon spiders, but that's not the case. They're actually most closely related to each other within the island, and then they just happen to come up with the same solution every time that they evolved on a new island, which is really incredibly cool. We also see a lot of diversity in the webs, in the web builders, and this has allowed them to diversify in their diet as well. And so some molecular diet work has actually showed that there's quite a bit of specialization in certain species, whereas we expect a lot of spiders to be generalist. The way that they shape their webs, the angle of the web, how high that they build the web, what plants that they build it on, the structure of the web itself, so how tight that webbing is, how many radii that there are, the size of it, all of that is actually very reflective of the species and then very reflective of the diet that they can catch, which is, again, super cool. These spiders, again, are coming out at nighttime, building their webs, and they take them down during the day. They recycle the silk and then they put them back up. They're very busy. So there's some cool stuff about Tetragnatha and their mating. So these are the long-jawed orb weavers, and they're so named because they have these very, very long mandibles. So when you see the chelicera and the teeth that are coming out, this is a very like signature. But they also have these. The males have this fashion on them that's a hook that allows them to hook onto the female. We'll see if this, I'll have to skip the YouTube commercial. So when you see the male and female come together here, you can see how they lock jaws. And then here's the hook right here that allows the male to kind of clasp onto the female. Now, part of the reason why he's clasping onto her is he's trying to keep away from her fangs. And then their bodies will come close together. And then the male can actually transfer sperm with his pedipalp into her epigenum. And so he's able to hold her in this mating position and then transfer the sperm to her. And this is how spider mating occurs. So if you ever want to know how to identify male versus a female spider, this is universally true. If they have these very swollen extra appendages, those are the pedipalps, that's a male. And then if they have an opening on their underside of their abdomen, that's a female. So color in spiders is really quite varied and really very, very cool as well. There are instances of structural color in spiders as well, but a lot of what we see is a result of laying down these pigments. And the pigments can then vary depending on if there's this other layer of these guanine crystals, because the guanine crystals, which are just a metabolic waste product, can make the spiders have this reflective, shiny, iridescent background. So while the yellow, red, and black pigments can combine into almost any color that you see, it's these guanine crystals that make them have that very iridescent shape. And spiders can also have variation in what they look like depending on what they eat. So this is a happy face spider that has eaten something that's green, and so it made it turn temporarily a very green color. Color polymorphism can actually happen within an individual, and so in this case, we're looking at Tetragnatha hawaiiensis, and it's using the green form to be cryptic against green leaves, and then it's using the dark brown morph to be cryptic against brown leaves. And so this can, again, vary within an individual. They can deposit, and sometimes it's related to age, sometimes it's just related to habitat variation. Tetragnatha and Nui is our rainbow Tetragnatha. Nui is named for rainbow in ʻElala, Hawaii. And they're remarkably diverse in color. So this is just 4 images. These are all different individuals. And that you might, assume if you were just basing it on color that they're different species, but this is all the same species, and this is found on Hawaii Island. Happy face spiders, we absolutely adore. I think everybody has a special place in their heart for these. They're quite small, they're not very big. And so, this is the species of happy face spider, but there's actually quite a few other Theridian species that are the close cousins of the happy face. And you see that there's some interesting color polymorphisms in these as well. And these are all different individuals, and they're all undescribed. So again, just a huge amount of diversity. With that within the Araneids, there's this genus Cyclosa. So this is a group that I've done a molecular phylogeny for, trying to understand what some of the undescribed biodiversity is. There has actually only been one person who's worked on this back in 1900. He described 5 species. A couple of those that he described, we actually weren't able to locate again. And so what we're going off of is somewhat just a new description of the entire genus. This is the daytime orb weaving spider in Hawaii, where the nighttime orb weaving spider is Tetragnatha, and they co-occur, and there's zero competition as far as we know, because they've divided up the niche space based on day and on night. And this genus there's about 180 species of Cyclosa globally. Here, we have 6 or 7 that we've found so far, but some of them are quite localized on, and again, single island endemics, one mountaintop. So Cyclosa pohole was found just in this region, where it's a very small body size. So for reference, the body sizes tends to be more like 15 millimeters. And this is a picture to kind of show you the blow up of this size spider. So this is on a leaf, and that's how small the spider is. These are trash line spiders. So just like in this image, they actually will put their trash in, and it's a way for them to avoid predation because they're in their web in the middle of the day. It's somewhat confusing for birds, which are their main predator, to be able to find the actual spider. So it's a way to hide. It's a camouflage mechanism. But in Hawaii, not every Cyclosa is actually using a trash line. An example of a very hardy trash line is Cyclosa that looks like turbinata, which is a mainland species. It's not turbinata. Again, it's not described yet. We're using web morphology to distinguish these ones as well, along with DNA and morphological taxonomy. But again, this is a group that's just from East Maui. This Cyclosa perkinsi is a species that's found in more disturbed habitats, more open or more mesic forest. And again, this one is actually found across the archipelago. Similarly, Cyclosa simplificata is found across the archipelago as well. So some taxa that are single island endemics, some that are quite broad in range. What's interesting about this is that there was a subspecies that was described in 1900, and when we investigated it, it was morphologically distinct, but not genetically distinct. But they do ecologically differ because they build their webs on these long bridges and threads across open gaps between the vegetation. So remember, webs can be a distinguishing feature for species. So perhaps this is sort of incipient new speciation. Another case of what looks like incipient speciation in this group is the Cyclosa tree species. So these ones are super cool. They have these like really flimsy, lazy webs, I like to say. And this spider doesn't take its web down every day and put it back up. So they seem, in this group, they especially seem to not care. But they build their webs right against the flat trunk of the ohia tree or koa tree, and they're only found right now, they're only found on Hawaii Island, and so they're kind of nested within that simplificata clade, and perhaps diverging again because of ecology. So we talked a bunch about these adaptive radiations of spiders, like Tetragnatha and Cyclosa, so an adaptive radiation where ecologically it's very important for how they're making their living. But there's also non-adaptive radiations, and one of the classic cases of that is the Orson Welles spider. So here's that giant sheet web that I was telling you about. So the sheet web can be quite large, and I've seen them build what I call condominiums, where there's like a whole bunch of sheet web spiders next to each other, like next to a big downed ohia tree, and again, a large spider. So it's non-adaptive because they're all kind of doing the same thing. They just have speciated on different mountains, and so they're not found as overlapping species in the same area. They're completely what we call allopatric, and they are also an interesting case because I told you that they're a case of island gigantism and they build these large webs. They have something called a kleptoparasite spider. The kleptoparasite is something that is actually getting all of its prey just by hanging out in like the upper reaches of the Orson-Welles web. So they don't make their own webs, they just kind of hunt from the garbage that's left over in the bigger spider. And we did a diet analysis on this using DNA. And in my lab now, we're starting to look at like spider diets and spider webs as ways to kind of sample the environment in a less destructive way. So rather than having to capture the insects and kill them, we can just do it by collecting environmental DNA. So of course we're trying to track biodiversity in this very changing world. And so one of the questions is like, what are the novel interactions, right? Because I told you that there are some spiders that are introduced, and this right now is looking at some of the taxa that have been collected, some of the genera, and then the plants that they're associated with, and this is where they're hunting, what their ecology is, etc. And if we overlay the non-native taxa, there's a huge number of novel interactions that are happening. And so we imagine that there's like competition or indirect competition, new predator-prey relationships. And so kind of uncovering this and understanding what those effects are is really important. So I will end there and take any questions. Thank you.
That was wonderful, Natalie. Thank you so much. I've never seen I didn't know we got that many spiders. It's amazing to see. Oh my goodness. So I know we have a few questions that have been submitted, and I think we'll first start with Camille. Let's see. Alrighty. Can you talk about any endemic bugs which have exploited non-native plants?
Yeah, so there are some examples, but it's much less than the native plant interactions, of course. So like ohia, I think, has more than 100 insects associated with it. And then for any of your native or non-native plants, that's going to be much less, if anything. But there are some notable examples. For one, there's the blackburn sphinx moth, which is a federally listed endangered species. It's a large hawk moth, and its native host was Nodosestrum, and those are very hard to find now. But luckily, it also feeds on the invasive Nicotiana glauca, the tree tobacco. And there you get into this kind of difficult situation where you've got this invasive weed that you want to take out, but at the same time you want this hawk moth to survive. So you have to kind of make a choice here. Are we just going to accept this weed at least in some places so that the moth can survive, at least until we maybe get the native host outplanted and back in healthy populations. But for the most part, there's only small examples where we know that native moths are using non-native plants.
Are there any cases for spiders, Natalie, where they're adapting?
Yeah, so actually this answers one of the questions in the chat too. That was about Theridian species that are the cousins of the happy face spider. And so we do see the native spiders that will use the Himalayan ginger. So you can find happy face spiders on the underside of Himalayan ginger. It seems like they're just using it as a broadleaf. So ilex is a really good place to look for native spiders that are these hiding during the daytime. So the green ecomorph of Tetragnatha, the Theridian happy face spiders, and their cousins as well. And then unfortunately, what happens with the ginger is that some spiders are able to use that habitat. So like the Philodoria spiders, the the running crab spiders. You'll find them there a lot, and they're very generalist. But the specialist spiders, the ones that are the web builders, that they're not going to be able to use ginger at all. So there's some winners and losers in that case, and yeah, so it's better for them to have other native broadleaf plants.
Yeah, definitely. It helps to have that diversity of different things to live on and eat upon. So yeah, that makes sense. Okay. For one more for Camille, is the bone collector the only carnivorous hyposmocoma?
It's not actually. So we have these different species groups, and the bone collector is in it on its own in its species group. So there must have been more at some point, but they've probably gone extinct, or we just haven't found them yet. But they're the sister group to the bone collectors is what we call the cigar case type, and those are all carnivores too. And Dan actually published about that some years ago. Is that some of these are snail hunters, so they actually hunt for the tiny native snails. So I mean, this is not like an action movie, right? It's going to be a pretty slow hunt, but there's a snail somewhere on a leaf, and then there's a caterpillar coming in, and it'll use its silk to pin it down, and then it'll go inside and straight up eat the little snail. But they'll eat anything really, like they'll eat each other too if one is bigger and one's a little smaller. So yeah, for how tiny they are, they're kind of ferocious little predators too. So in that group, there's about I think we're at about 30 species in that group, and they're on all the different islands.
Oh, so maybe one day we'll see a bone collector with a kahuli shell on top or something.
Who knows?
Okay, I'm gonna switch over to Natalie for a little bit. Is there a recommended way to mitigate Himalayan ginger in a way that's least impactful to native insects and spiders? Maybe this is for both of you, but yeah, is there any recommendations for mitigating insect damage?
Yeah, so I mean, there's a lot of this work that's been going on in the park to mitigate ginger. So removing it is the first problem, and because it's rhizomal, it tends to leave stuff behind. So really trying to get rid of it sometimes requires the application of herbicides. So clearing those herbicides out, and sometimes. So we've been interested in whether there is this like somewhat historic effect of where the ginger has been like cleared because you're applying chemicals in order to really get rid of it. But if you don't do that, it's impossible to dig it all the way out. You're just kind of getting the surface. And then restoration. So bringing back in the native plants is really what has to happen next, because otherwise then you just have kind of cleared the landscape, and then the next thing that's going to colonize is going to be one of these taxa that tends to move in very quickly to open landscapes. So yeah, just kind of planting what's good and removing what's bad.
Bit by bit. All right, we have one more from Chuck for Dr. Doorenweerd. I read in your paper that in captivity, the bone collector caterpillars will attack and eat any live, slow-moving, or immobilized insect prey, and they will even cannibalize each other. Is it known how they detect their prey? Through chemical, motion, combination of those?
No, it's not. So that's one of the interesting things that would be cool for follow-up research for sure. So we do know their vision is pretty poor. So it's probably not very much sight-based. So it's probably by scent. They do have scent organs. But what type of chemicals are exactly the cues that they use, we have no idea of yet.
So much to learn in the insect world. Let's see. So we know that the native bugs, that they thrive in the areas of native plants. So I guess, is there any final words before we end our webinar, as I guess any last words of wisdom for folks to do and to keep doing out there to conserve our native insects and keep learning more?
I'd just say plant native plants.
Keep planting, yes.
That was mine too. And try to reduce the amount of herbicides or insecticides that you use, because that does have a long-term effect on taxa. And we don't want to eliminate pollinators or the things that the spiders prey on, right? It's all kind of goes together. It's a big network of happiness.
Wonderful. Awesome. I loved it. I mean, I really, really thoroughly enjoyed both your presentations. I learned so much, and I know our audience really enjoyed it as well. As a reminder, this will be recorded, and we'll share it onto our YouTube channel as well as through our different networks. So thank you guys so much for participating, and I hope we can have you guys as speakers again in the future. So with that, I would just say mahalo nui.